Thermoregulation in mammals or humans, especially regulatory strategies. Mammals, including humans, maintain their high body temperature within a narrow range of about 36 to 38 degrees Celsius, and because of their endothermic characteristics, the origin of endothermy in birds and mammals have provided the opportunity to keep high metabolic rate and availability of energy round the clock. Thus has required greater ability to adaptations and has assisted in much of their wider diversity and distribution in diversified regions of the Earth. These regulate the rate of metabolic heat production, balancing it with the rate at which they gain or lose heat from the surroundings. The rate of heat production is increased by increased muscle contraction by movements or shivering, so-called shivering thermogenesis. Also, hormones trigger the heat production and as do thyroid hormones and are termed as non-shivering thermogenesis. Some mammals possess brown fat which is specialized for rapid heat production. In overproduction of heat, it is dissipated through exposed surfaces by increasing blood flow or the evaporating cooling. In mammals, it is observed that the skin has been adapted as the organ of thermoregulation. In cold temperatures, mammals have various mechanisms that regulate heat exchange within their environment. Vasodilation and vasoconstriction affect heat exchange and may contribute to regional temperature differences within an animal. On a cool day, a human's temperature may be several degrees lower in the arms and legs than in the trunk, where the most vital glands are situated. Most land mammals respond to cold by raising their furs, thereby trapping the thicker layer of stale air, and it acts as good insulator between animal skin and the surroundings. Humans mostly rely on a layer of fat just insulating beneath the skin as insulating material against heat loss. Similarly, marine animals such as whales and seals inhabit much colder water than their body temperature, have a very thick layer of insulating fat called a blubber just under the skin. In warm temperature, mammals dispose of their excess heat into warm seas by large number of blood vessels in the outer layer of skin. This dissipates the heat from the skin surface. In terrestrial mammals, in contrast, is a mechanism of evaporating cooling. The sweat gland activity and the evaporating cooling is one of the major temperature reducing strategies. Panting, the evaporating cooling in the digestive tract, is the other mechanism represented by dogs. Bats, etc., use saliva and urine for evaporating cooling. Thermostat function and feedback controls in human. The body temperature regulation in human is based on complex homeostatic systems facilitated by feedback mechanisms. The homeostatic thermostat is present in the hypothalamus, a brain part, and responds to changes in the temperature above and below a set point, which is 37 degrees Celsius. In case of increase in temperature above the set point, certain warm temperature sensitive thermoreceptors and skin, hypothalamus, and other parts of the nervous system send the signals to the system that increase the blood flow to the skin and also cause sweat gland activation, and the sweat is evaporated for cooling. In cold temperature, the cold receptors send the impulses to hypothalamus to inhibit heat loss mechanisms and activate heat conservation mechanisms. This include constriction of superficial blood vessels and stimulating shivering and non-shivering mechanisms. Temperature and fever. Pyrexia. In bacterial and viral infections, mainly leukocytes increase in number. These pathogens and the blood cells produce chemicals called as pyrogens. Pyrogens displace the set point of hypothalamus above the normal point of 37 degrees Celsius. Fever or high temperature helps in stimulating the protective mechanism against the pathogens.